Hi everybody, this is Michael Ralph, and today we're going to be setting up the Nitrogen Enrichment Lab. You're going to need everything you see right here, most notably a solution of mixed fertilizer and an original source of algae. The first thing we're going to need to do is get a sample of our pre-mixed fertilizer to work with at our group. I don't know how much fertilizer is put in here, but I do know that it's full strength according to what's on the carton. So I am going to call this 1 full X, 1.0x fertilizer, and I'm going to get a sample of it to take back to my group. I'm going to need 2 equal sized beakers. It's important that they're the same shape and the same size, and I'm going to pick a mark on one of my beakers. This is going to be my keep beaker. I'm going to pick a mark. 140 is easy on my 150 beaker. It doesn't matter how much it is as long as it is consistent and I can find the exact same mark on my other container. So I'm going to get some 1x fertilizer filled up to that line. And then I will head back to my group. Now that I'm back, the first thing I always want to do is I will always, whenever I have a full keep beaker, the very next thing I will do is cut it in half. So I'm going to take my keep beaker and I'm going to pour into my toss beaker until they are about even. That's pretty close actually. Now my keep beaker, you guessed it, I'm going to keep, so I will set that off to the side. My toss beaker is waste. That is going to go down the drain, but before I throw it away, I need to check and find out if I need anything out of this beaker. We're going to save samples as we dilute our fertilizer concentration. A 1x sample is an excellent sample to take, so I'm going to fill up to some designated line, and it can be again at the top of the label or you can mark it with a Sharpie, but some consistent volume from our toss beaker. There we go. And I can set that to the side after I've labeled it. And now that it's labeled, I can set that off to the side. Now that I have saved that sample, this can get thrown away down the drain. Now my toss beaker is empty again. The next step, my keep beaker, as we mentioned, we are going to keep this and I'm going to dilute this. So I'm going to fill this back up with water up to my original line. Since this was cut in half, so there is half as much of the 1x and we had diluted it with water, there's now half as much 1x. Half of 1x is 0.5x. So now what's in my keep beaker is half as strong as whatever was in my original 1x fertilizer. Check our list. If we need to save it, we'll have an extra step. But remember, first things first, we always cut it in half whenever it's full. Pretty close. And now we are back to our original condition. Do we need to save any from the toss? I'll look at my handout. 0.5 looks like it's not going to be saved, so I'm going to get rid of some 0.5 so that my toss beaker is now empty again, and I can fill it back up yet again. Back up to my original line. Now that it's been cut in half twice, what concentration do I have here now? Cut it in half again. Do I need any of this concentration? I suspect that you will, so we'll save from the toss. And now the rest of it can go down the drain. And we can continue this process as many times as we like, continuing to dilute and cut and save for as many dilutions as we need. So remember to continue to label each of your test tubes as you go so that you know what concentration is in each. And then our last step, once you have all your fertilizer concentrations, is we need to seed these with a beginning source of algae. So it doesn't matter, you can use a pond sample or a running culture, but it is important that you don't have any chunks, no hard big clumps of algae. We just want the algae water. And I'm going to get 1 milliliter of algae water to put into each one of these cultures so that that algae can start growing. So each one of our test tubes has algae water in it, and now that algae can start growing with each amount of fertilizer. From here, this just goes under the grow light, let it grow for a while, and we'll take our results here in a couple weeks. Good luck.